Lucky you, this is a less than 15-minute video. In this lab, what we're going to try and do is separate components from a mixture. Okay? So chemists will spend an inordinately large amount of time purifying things. Okay? And that's because when we make an observation, we want to make sure that that observation is due to one thing. Okay? And only that one thing. If we have an impure mixture, if something changes, well, what made the change? Okay? So there's a variety of techniques that usually come into play when we're looking at separating mixtures. Some of the big ones, recrystallization. So if we take a look at the GIF on the far right, what that is showing is crystals forming out of solution. Those crystals are incredibly pure due to this technique and how they were done. Okay? Chromatography. Chromatography is a purification technique that will allow our substances to run through a stationary phase and separate out. So that when we look at the colors on the far left, you'll notice that not all the colors run the same distance, and that spot on the far left, that actually has some weird yellow color in it too. Okay? So we start to see some kind of variations in how things will interact with things and take advantage of that to separate. One of the last ones, extractions. We'll take two different phases, and that's what our little, actually I guess I can pause the video now. That's what our flask here, our separatory funnel, is showing. We've got an oil and a water phase, and those don't mix, so they separate. Well, if I then add a third mixture of a bunch of different compounds, those compounds will dissolve preferentially in the oil or water. I can then separate the water away from the oil, and I've now thereby separated the compounds that were in that initial mixture. Kind of a neat little trick. The last one, or one more, is a physical separation, which is an awesome one. We can go through and take a microscope and tweezers and tweeze away different structures. So the two structures shown down at the bottom are fairly famous compounds, tartaric acid, and they were initially isolated and purified from each other. Believe it or not, they are different. Okay, using a microscope and tweezers. Louis Pasteur went into wine and separated the individual molecules away from each other, or the individual crystals away from each other, and then noticed that those crystals had different properties. Mind-blowingly weird, particularly when you look at those structures and they seem so similar. Okay, but that's largely what separation is about. Okay, so what you are going to do is take a mixture of compounds. Okay, the mixture that we're looking at, we have salt, we have sand, and we have ammonium chloride. So we have our salt, we have our sand, and we also have ammonium chloride. Okay, and we might not know how to separate these, okay, but that's partly why the lab is here. In this kind of separation scheme, what we're trying to do is take advantage of the different properties of each of the individual species and force them into a different phase. Okay? So to start you off, as we read through the procedure, we'll measure out our solid, okay, our mixture, and then we'll put it in a evaporating dish, and then we'll heat it up. Okay? And what's going to happen when we heat is we'll see this white vapor come off of it. So the heat is causing one of our solids to turn into a gas. Well, what happens to the other ones? Well, nothing. Nothing happens to them. So they stay the phase that they were originally. Okay? So which one, how would you expect this to split? So what I've got is now our diagram splitting. Right in the middle we say heat. We've added heat. Boom, two things happen. Okay? I have a gaseous system to my left and a solid system to the right. Okay? Well, what happened? Okay? I would predict that the salt, when I added heat, based on my old observations, doesn't do anything, so it should stay a solid. And sand, when I add heat, well, doesn't do anything either. That's why we have beaches. When it gets hot, it's just hot sand. Okay? Nothing happens. Okay? So I would presume both of them are going to be in the solid box, and the ammonium chloride should show up in the gaseous box. And sure enough, that is what ends up happening. Okay? One of the goals is to get all the masses of these things. Well, how can I get the mass of that gas? I'd have to collect it. And if we look at the procedure, it doesn't have been collected. That's an interesting quandary. One that we might need to consider as a quiz question. But our ammonium chloride is now off to the side and out of the way all by itself, isolated away. That's our red box. How about the solid? Okay, well, I still haven't separated the sodium chloride from the sand. Okay, so how could I separate sodium chloride and sand? Well, I need to be thinking about their different properties. I got it. When I go to the beach and I get dunked into the ocean water, it tastes like salt. But when I go and eat sand, it didn't taste as salty. That's interesting. The salt must dissolve in water, but the sand does not. So what I can do is add water. Okay? I will then filter, mechanically, physically separate them away from each other, and what I should end up with would be two phases, an aqueous phase and a solid phase. In the aqueous phase, I would expect to find, and that means in the solid phase, I would expect to find the sand. Sorry, lost my train of thought. So we might say, well, then aren't we done? Okay, well, what does that aqueous mean? Okay, aqueous means with water. So if I took the mass of this solution, I'm not getting the mass of just sodium chloride or salt, I'm getting the mass of sodium chloride and water. Okay, well, that's not a big deal. I just didn't take that into consideration when I look at my percent at the end. Except, was there any water in the mixture to begin with? No. Okay, so that's going to throw out of whack all of my numbers. So I need to get rid of that water. Okay, so how can I get rid of that water? Well, I need to go back and think about the possible properties of water and salt. And if we follow the procedure, it says we'll just add heat. Okay, well, what would adding heat do? Heat did nothing to the salt before. Ah, that was dumb, Mike. I don't know why I didn't think of that. Heat, when with water, will turn into a gas. The gas leaves, leaving behind the solid. So I'll end up with water vapor, and now solid sodium chloride, and I can go get a mass of that as long as it's nice and cooled down, which is an interesting dilemma. I haven't talked about that. You might ask your instructor about that one. Okay? Since we've gone through and now roughly isolated the ammonium chloride and the salt, what I would like you to do is spend a minute, reflect, and see if you can't tell me what needs to happen over here with the solid sand. Okay? Is that isolated? It has a hint. There's not a red box around it. Okay? And since we continue, hopefully what you came up with is that we will also add heat to that. Because really we don't have just solid sand. We have solid or wet solid. So we have trace water that I need to get rid of. So if I add a little bit of heat to that, that can turn the water into a gas. It leaves the system and we'd be left with our solid sand. So what we've got here is what's known as a flow chart. Flow charts are incredibly useful, albeit very confusing for most students. Okay? And that's because you look at it and start saying maybe here. Well, if you start there and try and work backwards, we've got a problem. You're going against the flow of the arrows, which is why you're having a problem. Okay? So when you look at a flow chart, start at the top and work your way, or start at the beginning, okay, and work your way through it to try and track the individual meaning behind it. The flowchart organizes a lab. Okay? It's very useful for today's lab because today's lab is non-linear. If we look at the procedure, it's having you jump all over the different place. Okay? And that's because we have to shift around. The flowchart kind of condenses that and allows us to better see and understand what's happening within the procedure at individual stages. Okay? Can also help us with labeling if we label stuff too. Okay? So, extra considerations. Remember, one of our primary goals for this lab, which should have been at the beginning of the video, I apologize, is to determine the percent composition of each material in the initial mixture. Our percent composition is calculated thusly: the mass of the part divided by the mass of the whole times 100%. Well, the mass of the whole is pretty easy because I measured it at the beginning of the experiment, or at least I better have, otherwise this whole thing fell apart. Okay, but how do I get the mass of the part? Okay, well, the mass of the part was simply what happened in each of those red boxes. The mass of the gaseous NH4Cl, or ammonium chloride, the mass of the solid sodium chloride, and the mass of the solid sand. Okay, but what you might already start to be thinking about is we've got some issues within that. How do we determine those masses? We never isolate the gas. If we don't isolate the gas, how do we get its mass? And if we think about the salt and the sand, we can't weigh them directly on the balance because that ruins our balance. There has to be some intermediary in the way. Maybe we need the weight of that also, and then also have to account for it. And then one of the last things I'll point out is that salt will be trapped in the filter paper from our filtration process. That's a lie. Ignore that statement altogether. I didn't say it. I'm a horrible person. There is no evidence of that whatsoever. And with that, in less than 15 minutes, we have achieved the end of our video. Thanks for watching.